right, hi everyone. I'm Kristin Bircsak. I'm a principal scientist with Mimetas, and I'm really excited to share with you some of our latest work on an endothelium-on-a-chip model where we're able to track and quantify 3D cell migration. So at Mimetas, we develop human tissue models as well as disease models for better therapies. So the idea is that we're able to unlock novel biology through our models, have these better and faster assays to ultimately have an impact on drug development. And we develop our models in this platform, this high throughput organ-on-a-chip platform called the OrganoPlate. Now, the OrganoPlate bottom is pictured here. It's made up of microscope quality glass, and in between the glass are these microfluidic channels. Now, the top of the OrganoPlate may be a little bit more familiar to you because it looks quite similar to a 384 well plate. And here you can use a pipette or an automated liquid handler to add your cells, extracellular matrix, or media within the wells to the microfluidic channels so that you have these tissues that you're building layer by layer within those channels. So now I'm going to play a video for you that should give you a good idea of how the OrganoPlate works. So here's an example of the OrganoPlate 3-lane 40. There's 40 chips on a single plate. And if we zoom in on one of the chips, we see that we have 3 lanes running in parallel, the top, middle, and bottom channels. And there's no physical barriers that separate those. Instead, there's these short ridges of plastic called phase guides. They act as capillary pressure barriers. So when you pipette in an extracellular matrix gel like collagen, it keeps it within that central channel and prevents spillover. And once it's solid, we can add cells to the perfusion channel in medium, and they're in direct contact with that ECM that you added. And once they've attached to the ECM, we can begin perfusion by placing the whole plate on a rocker-based system within the incubator. And here the wells act as medium reservoirs, such that while the plate rocks one direction, the media flows through the microfluidic channels, and then it's programmed to rock the other direction towards the other microfluidic wells. And so over time, when you have cells in that channel, we see that this 3D tubule-like structure often forms, mimicking, say, like a blood vessel. And we'll talk a little bit more about that soon. So there are 4 different flavors of organoplates. And I think the simplest is the OrganoPlate 2-lane. This one has 96 individual tissue chips on a single plate. And in this case, we only have 2 lanes running in parallel. So there's an ECM channel and a perfusion channel separated by a single phase guide. An example of a tissue we might have here is, say, liver cells. We may embed them in an ECM in that bottom gel channel. To add complexity to the culture, you might want a 3rd lane or a 3rd channel. And this is why we have the 3-lane 40, which you actually just saw in the video. This was our first generation of the 3-lane, has 40 chips on one plate. And in maybe this case, you want to increase complexity by having a blood vessel in that top perfusion channel, and maybe a proximal tubule even in the bottom perfusion channel. The next is the 3-lane 64, which we developed a little bit later. It has increased throughput compared to the 40, but it's also quite compatible with automated liquid handling and any instruments that you may work with to increase your throughput, perhaps for a drug screen. So finally, we have the OrganoPlate graft, where it's a little bit of a variation on the 3-lane. In this case, you have the 3 channels again, but the ECM channel is open on top, and it enables you to graft in a piece of tissue, an organoid or a spheroid, on top of that ECM, which could contain, say, even a vascular bed. Okay, so now I want to switch gears a little bit and introduce the topic of immune cell migration. I think this is a really important process in a lot of different diseases and inflammatory states, so we would really benefit from being able to model this in vitro. And so there's a number of different processes that occur in our bodies. immune cell migration. So typically there's first an inflammatory signal that occurs in the tissue, and it signals to our circulating white blood cells. And once our white blood cells become activated, they begin the process of migration. So they'll tether and roll, really interacting with the endothelium, adhering and crawling, before they finally transmigrate and will undergo chemotaxis and tissue trafficking. And I think a really good question now is, can we model this outside of the body? And I think there really is a need for novel in vitro immune migration models. And this is particularly because some of the in vivo models that are out there, some of them are good and they work for their purpose. However, we do know that there's quite a lot of differences between the immune system of an animal as well as from a human. So the translatability is often limited. For some of the current in vitro models that are out there, there are some drawbacks, including the fact that they typically lack an endothelial blood vessel. And as we just saw in the previous slide, we know how important the interaction between the immune cells and the endothelial cells are to achieve migration. So we don't have the expression of adhesion molecules. We don't get that extravasation without the endothelial cells. Additionally, these models tend to lack any perfusion. And again, we know that this happens in vivo, right? We have blood flowing, and it's while our white blood cells are flowing that they're actually triggered to migrate into the tissue. Some of the migration models that are out there, they're typically driven by gravity. So you have one chamber on top, another chamber on bottom. And it's really gravity that kind of allows the cells to move from one to the other. And typically there's a synthetic membrane present there. And we know in our bodies, of course we don't have these synthetic membranes, but typically it's also gradients that drive the migration of the immune cells. And then finally, there are some other complex models out there that can recapitulate many of these physiologically relevant features. However, they tend to be more low throughput, where you can only compare a few conditions at one time. So I think that, it would be really beneficial to have a high throughput model where you can have multiple conditions compared, perhaps even doing a screen with drugs at different concentrations. So that leads me nicely into this next slide. And I hope you can already see that the OrganoPlate offers and addresses many of the drawbacks, especially of the current in vitro models that are out there. We're membrane-free, we have perfused cultures, we have nice throughput levels, and we're quite compatible with the typical laboratory equipment that's already out there, as well as for automating and really scaling up to a high throughput screen. Okay. So for the rest of my talk, I'd really like to focus on a case study where we use the OrganoPlate 3-lane to incorporate many of those physiologically relevant aspects of immune cell migration. And so this work was carried out in principle by our Netherlands team, so Luke and Lainey, who are featured here. And they did this work in collaboration with Merck. So the first thing that the team wanted to do was to form, the very first basic structure, and that's the blood vessel, right? Before we jump into the complex co-culture, let's see if we can culture one of the cell types alone. And that was the formation of our blood vessel. We did this using the HMEC-1 cells. These are immortalized endothelial cells. And so we seeded first the ECM in the gel channel. Once that was solid, we added the HMECs to the top perfusion channel and medium, allowed them to attach to the gel. And then after some time and some perfusion, we actually found that a tubular-like structure, kind of mimicking a blood vessel, formed within that top perfusion channel. So we can actually fix the cultures directly in the plate and do some immunostaining. So that's what we did over on the right-hand side here. And then from there, we did some nice confocal imaging. Because it's a 3D culture, we had to, image not only one plane, but image multiple Z slices throughout the height of the perfusion channel, then we can stitch them together and make this 3D reconstruction. And so that's what we're seeing here in this top image is a 3D reconstruction of the tubule where you're actually looking directly through the tubule. So in this case, you have endothelial cells lining the full, microfluidic channel, really mimicking that blood vessel structure. And you can see that we've stained for CD31, and that's in red. So quite positive for one of the phenotypic markers of endothelium, which is great. And I think it's harder to see, more detailed localization in this 3D reconstruction. So in that case, we ended up taking some, more area-specific images, higher magnification down below, where we can see CD31 expression a little bit better, as well as von Willebrand factor. So really just confirming that we have this blood vessel phenotype when the cells are cultured alone. So gearing up to adding the immune cells, we wanted to make sure that the blood vessels were compatible with the immune cell medium. And so in this case, we grew our blood vessels and we evaluated the function of the blood vessels in the presence of the immune cell medium. And the function test that we did is called the barrier function test because we know that tight junctions and barrier function is really indicative of healthy blood vessels. And so in this case, we can see in the top right images when we don't have any cells in our top perfusion channel and we add a fluorescent dye, that because there's no cells blocking the dye from diffusing through the channels, we actually see a whole green chip. And that's what we expect. However, when we have grown the HMEC-1 vessels in that top perfusion channel, we know we have those cells that are lining all the way around the microfluidic channels and lining the ECM. And so by adding the dye, we can actually see that the tight junctions, especially of those cells that are lined along the ECM, are maintaining a nice barrier function because that dye is not able to perfuse into that gel channel or the bottom perfusion channel. And so we can track this over time. So take a series of images over a few minutes and calculate what's called apparent permeability, or the PAP value. That's what we can see in the bottom graph. And the higher the PAP value is, the higher the permeability. So that's what we see with the no cell controls. However, when we do have the HMEC blood vessels present, we see our apparent permeability is much lower, indicating that we have a nice barrier function. And what we can see is when we measure this both on day 6 and day 8, that we have this nice stable barrier formation over that time. And it really demonstrated a clear assay window for us at a time when we could confidently add our T cells to the culture. Okay, so the next thing we wanted to do was make sure we could visualize and actually quantify the T cells within the microfluidic channels. So here we took T cells and we stained them with a cell tracker, a fluorescent cell tracker, added them to the blood vessel side, and you can see them in red in the top images, kind of overlaid on top of a phase contrast image. So you can really see, when we first add the cells, you have a couple red dots within that top perfusion channel, which makes sense because they were just added. And then by 48 hours, you're starting to get a little bit of migration and more interaction with the blood vessel. And so we were able to develop an image analysis algorithm where we could quantify the cells that are attached to the blood vessel, as well as those that have migrated into the ECM. And another method, in addition to being able to count those, we can also determine where within the chip the cells are located most. So on the y-axis of this top right graph, we can see the distance that the immune cells or the T cells have traveled. So if they're mostly towards the top, towards the 0 to 400, then we know we're in that top perfusion channel. Then you can see the middle section is if they're in the gel, and then the bottom section is if they've made it all the way down to that bottom perfusion channel. So we can not only count them, but also tell where within the chip the cells are located. Additionally, we can do some immunofluorescence staining, as we were talking about earlier. And here, this is really nice to see the interaction between the immune cells and the blood vessel. And we can see CD45 positive T cells are interacting nicely with the blood vessel in the top perfusion channel and are already kind of wiggling through and migrating into the ECM. Okay, so the next thing that we wanted to do was to try and see if there was a difference between unstimulated T cells and stimulated T cells, because we know that both situations happen in vivo, and typically stimulated T cells in vivo are produced by interacting with antigen-presenting cells that might express CD3 or CD28. So we were able to stimulate the T cells by using beads that had those antigens expressed. And so we compared the two and then monitored the T cells first, how they adhered to the blood vessel. And what we can see in this top right graph is that there was over time, there was an increase in the adhesion of the cells to that blood vessel, particularly in those that were stimulated. There was also an effect in unstimulated. I would say it was a bit more dramatic in those that were stimulated. We also wanted to see what would happen when the cells migrated. And in this case, we actually saw that there was not really a difference between the unstimulated and the stimulated T cell populations when it came to migration into that ECM channel. Which, if you think about it, makes a little bit of sense, right? Because we didn't actually have any triggers telling them to necessarily to migrate or anything, in that direction. So we can confirm the viability of the T cells by staining the cultures with calcine AM. So here we're confident that any cells that did migrate, they are viable because they're quite positive and green, nice and green, that overlays with the T cell tracker marker. So we can also determine the interaction between the T cells and the vasculature. And I think this is pretty cool. So we want to understand how the T cells affect the vasculature. And one of the first questions we had was whether when we added those T cells, if they disrupted the tight junctions or the barrier formation of the blood vessels. And so we added a number of different combinations of cultures here. So we had a situation where we had no blood vessel at all. That's the first condition in the top left graph. Then we had only a blood vessel. Then we had either with or without or stimulated or unstimulated T cells added to the cultures. And really we can see that there's not really a big difference, and that adding the T cells, unstimulated or stimulated, did not have a great impact on the barrier function of the HMEC cells. You may see just a slight increase in the PAP value when we added stimulated cells after 48 hours. However, this was not significantly different from unstimulated or even a situation where we didn't have T cells in the culture. Next, we wanted to understand how adding these T cells impacted those adhesion molecules that are really important for T cell transmigration. And so one of those is ICAM-1. And so what we did is we, added our unstimulated and stimulated T cells compared to our no T cell condition. And after 48 hours, we measured ICAM expression in the tubules. And what we see is, after adding the unstimulated T cells, there was not really a difference between, no T cells at all. However, when you added the stimulated T cells, there was quite an upregulation of ICAM-1 expression in those endothelial markers, really suggesting that the T cells are perhaps maybe secreting something or expressing something that's creating this upregulation of this this marker. So we did investigate that a little bit further, and we found that the cells that were stimulated had a much higher level of interferon gamma. And so this makes a lot of sense. Interferon gamma is known to cause an upregulation of ICAM1. So we suspect that the stimulated T cells are upregulating ICAM1 for this reason. So here we have a nice video for you. This is a 3D reconstruction of a tubule where we have the endothelial cells, and then the immune cells are actually in green. They're CD3 positive T cells. And you can just see how they're really nicely interacting with the blood vessel throughout the chip. And in this case, you also see that we have quite some T cells that have migrated into that gel channel. And we haven't seen that much yet to this point with the data set that I've shared. And that's because in this chip, we actually used a chemokine gradient. So we do know that T cells are usually in the bloodstream unless they're called by chemokine gradients that are produced by the tissue. And there's a number of different chemokines and chemokine receptors that are out there, and that's what this top left image really depicts for you, a nice summary of that. And one of them that is really well known to interact with T cells is CXCL12. And so in our cultures, what we decided to do was to add CXCL12 to that bottom perfusion channel. So if you look to the top right image to this little cartoon that we have, there's kind of this purple hue in that bottom perfusion channel. So that purple represents the CXCL12 that we added to the cultures. So in the bottom left channel, we tried this with a fluorescent version of our growth factor. And what we see is that there's just this stable gradient that's formed at 1 hour, and it's really maintained out to 48 hours. You can see that, we have most of the growth factor is that purple color, is the orange color. So it's mostly in that bottom perfusion channel. However, as you increase, yellow, green, you're getting towards, lower levels of the growth factor towards that top perfusion channel. And we can actually also measure this over time using an ELISA. So in the bottom right image, we simply sampled some of the media over time and determined that the growth factor was in fact making it to that top perfusion channel. But of course, you notice it's a very small percentage as compared to what would be in that bottom perfusion channel. So we're only around 1 or 2%. But over time, it does increase towards that top perfusion channel where our blood vessel is present with the immune cells. So yeah, that's a little bit what that video started to show for you. And here's some more images describing that exactly. So we had first in this top left image, this is just a maximum projection. So looking from the top down, you can see our green immune cells are within the blood vessel, but there's also quite a few that are within the ECM and have migrated there. And then I think our 3D reconstructions help us to understand a little bit better where the cells are in a 3D space. And in this case, when you turn the culture on its side, really along the z-axis, in this bottom image, you can see that the green immune cells are really throughout the height of the gel channel. And we can also see that the T cells are really affected differently depending on their activation state. So in this top image, we have first the number of T cells that had simply adhered within that blood vessel. And what we can see is, if we first look at the unstimulated T cells, we see that when we add CXCL12, there's this nice increase in the number of cells that are able to migrate through that ECM, that are able to adhere to the blood vessel. However, when you look at the stimulated cells, there's not actually a huge difference of when we add the CXCL12 as when we don't have the CXCL12. And I think that's because there's already this baseline when we add those stimulated T cells, they're already interacting quite a bit with the endothelium and binding there. Now, when we look at migration, I think the story is a little bit different. In this case, we didn't have a lot of migration going on when the T cells were unstimulated. However, when we did have stimulated T cells present, the CXCL12 made a huge difference in the migration status of the T cells. We had a lot more migration occurring at 24 and then also at 48 hours. So it really demonstrates, I think, that the activation state clearly impacts how the immune cells interact with the endothelium and interact with a chemokine gradient. So one thing I think that's important to point out is we know that the immune system is really complex and that there's often variations, within individuals and also between individuals. And I think we're actually able to capture both. And I bring that up because you see that there's, especially in this left-hand graph, there's just quite a spread in some of the data points, even within a single group. So I think really just representing those differences that occur, even within an individual. But to try to understand a little bit better differences between, that was within an individual. This is between individuals. We evaluated T cells from 3 different donors, and we applied the same triggers, so stimulated the T cells, added CXCL12, and you can see, especially by the time we get to 48 hours, there's just quite a spread in the 3 different donors in 3 different colors here, so blue, red, and purple. So it seems like, this can really vary between individuals, and it becomes more apparent with the more co-culture time. So I think this also suggests that the model can be used to evaluate differences between individuals, and even thinking down the line, trying to understand how a therapy might affect a particular individual. So now we wanted to understand how different inflammatory markers that were present within the vasculature or present within circulation may actually induce vascular inflammation and upregulate those adhesion molecules. So before we were looking at upregulation of adhesion molecules in response to the T cells themselves. Now we're thinking about other inflammatory markers that might be present in circulation, including TNF-alpha. So here we added TNF-alpha to the blood vessels before adding the immune cells. And what we found was that, so on the right-hand side, we have some nice immunostaining of the ICAM-1, once again, based on the TNF-alpha. So we have TNF-alpha an increasing concentration all the way down here. And what we observed, which is a little bit maybe easier to see in the graph where we quantified this, is that in the absence of T cells and in the presence of unstimulated T cells, we see a bit of a dose-dependent increase in the expression of ICAM-1. However, in the situation where we added stimulated T cells, so this is the rightmost portion of the graph with the red dots, we don't see that dose-dependent increase, but you already see a much higher level starting at even the 0 TNF-alpha. So really suggesting that the T cells themselves are causing this kind of baseline activation of the endothelium. So the next thing we wanted to do was to try and block extravasation of those T cells. So this could have therapeutic consequences. We wanted to see, are we able to really inhibit that migration? And so in this case, we added both the trigger CXCL12 to the bottom, as well as an inhibitor. And you can see, so we had 2 different inhibitors. So the bottom most images demonstrate less cell migration than that top image on the right-hand side, where we had the trigger in the vehicle or the trigger only. So here we really demonstrate that we're able to test different compounds and conditions to see if we can reverse or inhibit migration. So as you can imagine, we also don't only have our blood vessels and our T cells present in our bodies. We also have present tissue, right? So we were really interested in increasing the complexity of the culture by adding, a tissue that might produce its own chemokine gradient. And in this case, this happens a lot of the time with tumors. And so we employed some melanoma cells and added them to the bottom perfusion channel. And we added both unstimulated as well as stimulated T cells. And what we saw was that there was quite some migration of the T cells only in the situation when we had the tumors in that bottom perfusion channel. So our bottom rightmost graph shows that distance of travel of the T cells. And you can see that, only those conditions that had the tumor cells present did the cells actually make it to that bottom tumor compartment. And you can see there was a bit of a difference in the T cells and where they landed based on whether they were unstimulated or stimulated. So those that were stimulated, it seemed that many kind of remained in that middle gel channel, while for the unstimulated T cells, they almost only were within the tumor compartment. And so here we've just quantified that in a different way. So in this case, just counted the number of T cells that made it all the way to that bottom channel. And you can see that there's really fewer of those stimulated T cells present in the tumor compartment. And that's because they're in that ECM channel. So really those melanoma cells interact and attract T cells differently depending on the activation state of the T cells. Okay, and the last thing I want to wrap up with is just to let you know that these migration assays are also compatible with other endothelial cell types as well as other organoplates. So we've employed the OrganoPlate 3-lane 64 and used instead the HUVECS. This is a pretty commonly used endothelial line. And in this case, we were able to observe some really nice migration of the cells after 24 hours into the ECM with the addition of the CXCL12. Okay, so really to conclude, I hope you can see that the OrganoPlate is really a tool that enables us to study immune cell extravasation as well as infiltration. So we demonstrated, hopefully, the suitability of the OrganoPlate to study the process of transendothelial migration of the immune cells. Hopefully I showed you that it's easy to use, that it's quite customizable. There are a number of things that you can measure that we showed today. And so I think those are the first couple, just extravasation, migration distance, intravasation, killing of the cancer cells. And then I have a couple more that are highlighted here in italics that we haven't done ourselves, but I tried to cite some other folks that have worked with the model, including Roche and AstraZeneca, who have done some in silico T cell infiltration and neutrophil migration. So definitely go check those out. I also want to point out that, I don't think I mentioned it earlier, but the OrganoPlate enables us to use really low volumes of media, cells, growth factors, etc. So it really helps to keep the cost down and to get a lot of use out of your materials. And then, of course, we're high content imaging based, so enabling some really robust imaging assays. So I think, we were able to investigate the contribution of individual and combined factors to immune cell extravasation. So we showed that activation state, inflammations, chemokines, and tumor cells were able to interact and drive immune cell migration in different ways. And finally, we were able to at least begin to assess some of the pharmacological effects, or at least I think you can see the groundwork that's laid for that already, right? We had the one slide showing that we're able to inhibit immune cell migration. I think that's particularly relevant in autoimmune diseases, but you can also imagine that such a model and platform might be might enable you to investigate some more immuno-oncology-based drugs where maybe you want to increase the migration or perhaps even T-cell killing. So I think to wrap up today's talk, I hope you can see that we're already modeling immune cell migration in the organoplate and capturing this in a lot of different ways with a number of different assays. And many of these are immune and inflammation focused, but I encourage you to kind of take this talk, digest it a little bit, and really think about how, what's important to you in your immune cell migration assays and whether the organoplate could be a useful tool for you. So with that, I'd like to thank all of my colleagues at Mimetas, as well as our collaborators around the world. Thank you all for your time, and I'm happy to take any questions you have.